Nucleic acids, the structure and function of DNA and RNA. We're going to look briefly at the way nucleic acids work and how everything is put together. This has relevance whether we're looking at plants, whether we're looking at animals, whether we're looking at bacteria. There may be small differences between each one of these, but these differences are not significant. This is your genetic code, and it's an interesting thing because this is your genetic code that works on all organisms. The only difference between what we've got here and what happens in bacteria is the start codon, because instead of coding for methionine, it codes for formethionine, which is a little bit different, but the rest of it all works together. And, we could spend a lot of time talking about it, but that's not important right now. What we're looking at is we're looking at the different materials in here and how all of this is coded for by the DNA, the messenger RNA, and the transfer RNA. On the messenger RNA, we have these things we call codons, and the codon is the three little bits of information where it would be three base pairs. You've got on the left-hand side, you've got your first base pair. On the top, you've got your second base pair. On the right-hand side, you've got your third base pair. And you can see how this information allows us to code for all of the amino acids that normally go into proteins. This becomes a very interesting thing to look at because it is our genetic code, and these are called codons. The codons are found on the messenger RNA. You find the corresponding anticodon on the transfer RNA, and then that information can be passed on into the polypeptide. So if they take a protein that they've found and they sequence it and they find out what are the amino acids in it, they can then tell what the genetic code was that wrote it. Then they can go back and look for that genetic code in a chromosome, and when they find that, they know exactly where the gene is found. And this has become very interesting because this is what we do nowadays, now that we have completed the Human Genome Project. The genetic code there is used in RNA transcription. This is RNA transcription. And in RNA transcription, what happens is the place where the genetic material is that gives us the information is unraveled. There is an RNA polymerase, which is an enzyme that comes in, it grabs hold of the material, and it starts moving down this line, creating it what we would call an RNA transcript. And the RNA transcript pairs up the C's and the G's, and the T's and the A's, and the A's and the U's. It then gives us one of the four different types of RNA that we know of. These RNAs will then go out to the ribosome where they will be transcribed to produce a final product. Now let's see how that works. The way a ribosome functions is like this. You see this particular thing, and this is a kind of a very interesting representation. And in it on the bottom, you see the messenger RNA, which is this big long strand, and everything is broken down into these little groups of threes, which are called the codons, and the codons go back to that segment that we looked at, where we've got our genetic code in there, and it starts off going down reading until it finds the AUG material. Once it does that, it brings a transfer RNA in, grabs hold of it. The transfer RNA will then have its amino acid. It will grab hold of the amino acid, the transfer RNA. It will move it from the A site to the P site, then bring in a new transfer RNA, grab the amino acid off of the old one, bind it to the new one, and it moves along like that. And as it does that, it creates this new peptide or this new protein. So over here, we see various parts of the ribosome. Bottom part is the small subunit. Above that, we have the large subunit. The two of those come together and they make the ribosome. And they do that once they find the proper piece of DNA. In the center of it, you see the A site and the P site, where things are actually read, the transfer RNA is brought in, and then the amino acid is taken and passed from one onto the next. Here you see the A and P site, where the amino acid is transferred onto the protein that is being produced. Once the amino acid is taken from the transfer RNA, the transfer RNA without the amino acid is released, which means it goes back into the cytoplasm to find the proper amino acid to bring that in. And over here you can see the new transfer RNAs with the proper amino acids on them being brought in. Up here you see the protein that is being formed through this entire process. The messenger RNA travels in one direction, because the ribosome travels in the other direction. So in order to make a protein, it takes all of these pieces and it takes all of this going together in order to be able to make this protein. So they are little kind of readers where they read the material and then they create this particular protein, and then that particular protein would be released off, or that polypeptide, as we often call it, would be released off. And this is the way the ribosome works. Therefore, when we look at DNA, this is the way DNA comes together. These are the various nucleic acids that are important in the DNA segment. And this is how DNA is used, and from it we get a product, which is the information. These proteins that are produced then have the function of either being an enzyme or they have structural functions, and they are what makes everything function the way they do.